I just wanted to sort of start off with, we don't do most of our work on pandemic preparedness, but it's something we've spent some time thinking about. And this is a non-flu example, but why it's so hard to predict pandemics and indeed large infectious disease outbreaks. This is sort of a classic example of a large coupled problem, where the data graph on the upper right, your upper left, is Ebola infections. And I think most of us who spent time thinking about this thought it would continue to bump along with a few tens or maybe hundreds of infections per year. And so it did until not too long ago when you saw that spike that has now since come down. And that, we really don't know whether that was completely due to human factors, due to viral factors, due to ecological factors, due to pure stochastic elements. What in this very large complex system caused a great increase in disease transmission. For flu, there is a history of pandemics, and the one virologic factor that is highest confidence for positive predictive value for pandemic transmission is the hemagglutinin coat protein. And a lot of the work in my group focuses on physical interactions in cell entry by influenza. And what's wonderful about flu as a system is that there are fairly long-standing mutational data that show that as the virus gets taken up into endosomes and acidification of the endosomes triggers conformational changes in the viral coat proteins, that this fusion apparatus here is analogous to the fusion apparatus in synaptic exocytosis and a lot of other viral systems as well. But for flu, there are mutational data suggesting that you can change the membrane-inserted parts of either of these and either stop fusion halfway or block it altogether. That it's not purely a proteins as clothespins model and they pull the membranes together and magic happens, but that protein-membrane interactions are critical to the success of viral infection. And so for those of us who care about membrane biophysics, that makes this a really wonderful problem. What makes this a bit of a hard problem, and you actually see a very gentle movie on the lower left of single virus fusion events from our group, is that infection is very heterogeneous on a single virus level. That if you look at a histogram of fusion events versus time, that the timescales for this are seconds, they're long, and that if you were to even be able to reach this timescale, having a sort of single event movie for one virus fusing wouldn't really tell you much about the outcomes of this process. That they're sufficiently heterogeneous that you really need good statistical sampling, and indeed that's what's really required on the experimental level. So this gives us a bad combination of factors from a sort of computational viewpoint, that because of heterogeneous outcomes, we need many simulations for good statistics. Because the decorrelation times in these systems are slow, as well as the overall waiting times, we need fairly long simulations. And because even the most simplified sort of full-scale systems are several million atoms, we need fairly large simulations. And then finally, this is sort of a classic example of biological effects where some fairly subtle perturbations can cause substantial differences in the biological outcomes. So we need relatively high fidelity simulations, at least in key areas. So this obviously is enough to keep a lot of us busy for a long time. And a lot of my work focuses on unraveling these virus membrane interactions surrounding fusion, preceding fusion, during fusion. And we try to ask the question of what are the relevant physical interactions that control both the mechanisms and outcomes of fusion, and try to build an integrated understanding using both large-scale simulations and biophysical experiments. Today I'm going to tell you about some work actually preceding some of our Blue Waters work on membrane interfaces leading up to fusion, and then some work from a Great Lakes Consortium award, so a relatively small-scale Blue Waters award on membrane protein interactions involved in fusion. We take a multi-pronged approach because of the challenge of these sorts of systems. We do some simulations on what I sort of call mid-scale systems, not trying to say, let's do the whole thing, but trying to say, what do we think might be the smallest unit that contains all the relevant pieces? And those are several million atoms, and proceed over fairly long timescales. We also do a lot of work trying to relate more closely to some specific biophysical observables, where we look at isolated components or combinations of components, looking at questions of membrane heterogeneity, changes to membrane composition, protein-protein interactions in the membrane on what are these days not trivial but relatively modest-sized simulations, several 100,000 atoms. One thing on some prior leadership class computing allocations that we observed is that when two lipid bilayers come together prior to fusion, that they can form a stable membrane-membrane interface that can persist depending on the system. On the left is a vesicle-vesicle interface from some work that we published about 5 years ago. And there, these could be stable for as short as tens of nanoseconds, for as long as we could sample. On the right, you see a similar interface brought about by a full flu hemagglutinin assembly. We ran several simulations for several microseconds, and these interfaces could be stable for long periods of time. This was highly unexpected. This has since been at least indirectly supported by experimental evidence, some of which is published, some of which will hopefully be coming out shortly from other groups, suggesting that something strange is going on at these interfaces. When we went a little bit deeper, we noticed that at these membrane-membrane interfaces, water mobility decreased markedly, although this was still essentially a solvated interface. This was not a dry interface. This was not a liquid-vapor transition, but remained liquid water, but very slow. We've since gone on, and this was actually using some resources at Oak Ridge, to characterize the dynamics of water between lipid membranes. And we've been able to show that part of why just trying to hammer these sort of long fusion simulations was not working particularly well is that when the two membranes come close together, that the water dynamics can become glassy. And we can show that via sort of rigorous persistence and exchange time measurements using the physics of glass formers. And this work has recently been published in JAX. And I sort of wanted to show this as an example of where we started just saying, let's take what we think is our system of interest, push it as far as we can, see what we can get. We didn't get to where we thought we were going. We got stuck halfway along the way, and there was some interesting physics going on. So that's been, I'd say, a good story of making lemonade. And we're still, we think we have a sense of what's going on here. We now have some tricks to essentially use still unbiased simulations, but some sampling approaches to get past these transitions and sort of see what the next major barriers are. One of the approaches that both these sorts of large problems and what I'm going to tell you about next that we've been running on blue waters really require is multi-level parallelism. That, again, these are fairly large systems, and we want each individual simulation partition to scale over large numbers of CPU or GPU cores. But we also need parallelism between individual simulation partitions. Whether a lot of the work that we've done to date on blue waters has been fairly ad hoc, that we have scripts to launch jobs, we have scripts to run analysis, do sampling, launch more jobs. We've also, in collaboration with Eric Lindahl, developed a DAG engine to coordinate this in a domain-specific language, sort of a data flow language that's called Copernicus. There are a number of other similar approaches that really allow relatively fluid interchange between simulation workers that can each then run on large numbers of cores as needed. And so I think this is something that, whether this solution or something related, is very much a need in our community and somewhere where I think a lot of us are going, that as we have different sorts of sampling strategies, potentially different sorts of biasing strategies, as we're running not just one large coupled simulation, but maybe 100 or more. I've done some things where we've had 1 million jobs in flight at a time. And when you do that, you really can't just be playing data jockey yourself. That systematic methods for this really become required. So the system that we've run most recently on blue waters is an isolated domain of flu hemagglutinin, and often not the one that most people think about. That this is the transmembrane anchor of the fusion protein in the viral membrane. So people often think about sort of the business end that gets stuck into the cell, or the soluble portion that does the work of pulling membranes together. This is the viral anchor piece. And what's kind of interesting about it is, again, in the 90s, there were a set of mutational experiments where this is where truncations, either partial or complete truncations to this domain, could arrest fusion, suggesting that something going on here is required for the fusion process, that if you just put a lipid anchor on the rest of the fusion protein, it's not sufficient. And it's, since the soluble domain is trimeric, there are at least 3 copies of this transmembrane anchor in close proximity. There may be more, because multiple trimers likely act together for optimal fusion. So we asked the question as to whether these transmembrane domains could stably interact, and then ultimately we want to ask whether these interactions could play a role in the fusion process, and if so, what might that be? To do this, we've taken a multi-resolution approach, essentially to use coarse-grained simulations to sample diffusional processes, and then to examine many different encounter complexes and use those to spawn off atomic resolution simulations to sample the conformational equilibria of those encounter complexes. So this is, again, not a, we're not spawning atomistic simulations of every single possible element of these, sort of snapshots of these simulations as isolated TM domains are diffusing around. But once there are encounter complexes, that's where we're sort of engaging the statistical sampling to try to get at the atomistic dynamics of, are these complexes stable? What do they look like? How much variability do they have? And just very briefly, our findings thus far are that when we run about 24, 25 different coarse grain simulations from different starting conditions, that in our simulations, all of the transmembrane domains do form stable trimers. And then when we cluster those simulation snapshots and launch for a given sequence, we launch about 50 atomic resolution simulations, that all of those do stay stably associated. We can look at the intermonomer contacts, and they show some plasticity, but seem to show a fairly common pattern of contacts of these staying primarily in register. So this was interesting. There were some indirect experimental data in support of this, but no one really knew that, that we'd find this. What we then went on to do was to identify the four most likely or most frequent contacts between these trimers and mutate all four of those. And when we mutate all four of those, interestingly, the trimers stayed very stable. And that's shown here, that this is sort of the peptide center of mass for our starting configurations prior to coarse grain simulation. This is where we start the trimers, and essentially you see that everything remains within there, very stable, that the four contacts that we disrupt go down in frequency, but that, thank you, other contacts in their vicinity go up in frequency, so that there's some plasticity in the association of these monomers. And this is indeed very consistent with experimental data, that a number of point mutations and even pair mutations to the transmembrane regions, as long as everything can get folded and inserted, is still permissive of fusion. So we have this very interesting system where getting rid of it altogether destroys fusion, but that even taking what appear to be the most stable contacts and abrogating those seems to have no effect on the stability of the complex, and also seems to have minimal effect on the biological outcomes. So that's something we're going on to investigate further, and we're very interested in what role this then may play in the fusion process. So sort of the larger overview of this work, where this particular project fits in, is that we're interested in building an integrated understanding of flu viral fusion, where we both do methods work to build integrated statistical models at a simulation level using high-level parallelism, where we're then sort of composing this of both smaller scale protein membrane dynamics simulations, larger scale models of flu-mediated membrane fusion, integrating that with biophysical experiments, largely using fusion as an outcome, also some structural work that we're doing. And then ultimately, if you start thinking about, okay, what is the space of possible mechanisms given all these sorts of different simulation inputs, given all these sorts of experimental inputs, this starts to sound like a very large, high-dimensional statistical learning problem. And indeed, that's the way I like to think about it. It's a highly underdetermined problem, but I think trying to figure out what are the ways to narrow this down and to direct further experiments and simulations based on uncertainties in the large-scale problem is something that interests us a lot. So with that, I'll wrap up. The work I talked to you about has been done, the methods work by Sander Prank, who was a postdoc technically with Eric Lindahl. Eric kindly lent him to me for a lot of this collaborative work. Matt Eckler in my group and Bob Raleigh have also done the work I've shown to you. We're grateful for funding in terms of money, for funding in terms of compute resources, and thank you for your time.
All right, thanks, Peter. All right, time for maybe one question. People look a little sleepy out there. So workflows always seem to be interesting. So there's Copernicus, Kepler, Pegasus, Swift. I don't choose that.
Okay.
What's your feeling on how we're doing with the number of rapidly expanding?
I think, I feel to a certain extent we're at a stage where I'm inclined to sort of let 1,000 flowers bloom. That there are a lot of application-specific needs that are sometimes hard to engineer into more generic workflow tools, at least in their current state of maturity. And what I think to some extent, on the one hand, people who build generic workflow tools want to, and indeed probably are, spending a lot of time trying to adapt them to the needs of different use cases. But I think building sort of more domain-specific workflow tools is also useful. And I think that also the real challenges are things like, what are the requirements of your problem? That some things require certain kinds of latencies, some require certain kinds of scaling.